Hello everyone, welcome back to Dr. K. Primer lecture series presenting by Dr. K. Prem, that's me. Today we will talk about cloning vector, that is PBR322. So before going to discuss about PBR322 features, I just want to tell you what are plasmids. Plasmids are covalently closed circular extrachromosomal DNA molecule. Covalently closed circular extrachromosomal DNA molecules. They have their own origin of replication for autonomous replication. So plasmids do not depend on genomic DNA for replication. In addition to the origin of replication, plasmids also carry genes which are very important to adapt to the existing environment. They also carry the elements which are required to equal distribution of plasmids into the daughter cells. There are different types of natural plasmids reported from both prokaryotes and lower eukaryotes. So natural plasmids are used to construct a artificial plasmid. So PBR322, which is first artificial plasmid used for cloning of DNA fragments. So this one is constructed by using the natural plasmids, right? So this is a PBR322 is a cloning vector. What is a cloning vector? Means whatever gene of interest or gene of interest or a DNA fragment can be cloned into the vector by using the restriction endonuclease and other enzymes. And that fragment of DNA you cloned can be amplified in the E. coli. That's what is in vivo amplification. So more copies of cloned gene can be obtained through the vectors, right? So in today's lecture, we will talk about what is PBR322 and structure of PBR322 and the 10 features of PBR322 and advantages or limitations of PBR322. This is the backbone of this is the PBR322 whose size is 4,363 base pairs. This is the origin of replication. And the second element is repressor of primer, which controls the copy number of plasmid. Third element is TC, tetracycline resistance gene. The fourth element in the PBR322 is Ampicillin resistance gene. It is having four important components. One is origin of replication, a repressor of primer, and tetracycline resistance gene, and the fourth one is ampicillin resistance gene. Then now we will look into the what are those functions. Origin of replication is necessary for replication, autonomous replication, and repressor of primer is required for or regulates the copy number of plasmid per cell. Tetracycline resistance gene, that's nothing but TET-A gene, which encodes a transporter molecule which exports the accumulated tetracycline from cell to outside so that cell can survive in the presence of tetracycline. The next one is ampicillin resistance gene is nothing but beta lactamase. Beta lactamase can act on the lactam group of penicillins, which can break down the lactam ring and converts the penicillin into penicillinic acid. So whenever a vector which is consist of ampicillin resistance gene, that's a beta lactamase, that vector containing host can grow in the presence of ampicillin because the beta lactamase can degrade the ampicillin or penicillin, which inhibit the cell wall synthesis. So these are the important features or elements of the pBr322. And coming to the most important one, unique restriction sites. There are 29 unique restriction sites present in the PBR 322. 29 unique restriction sites are there. But 5 restriction sites are present in the ampicillin resistant gene, PST1, PU1, SCA1, BSA1, ASC1. When it comes to the tetracycline resistance gene, in that there are 9 unique restriction sites are there. They are EcoRV, NHE1, MAMH1, SPH1, SAL1, SMA1, NRE1, CLA1, and INT. So total, out of 29, 14 restriction sites from both ampicillin resistance marker and tetracycline resistance marker we can use for cloning. Right? Out of 29, 14, 9 from tetracycline gene, which are there in the tetracycline gene, and 5 from ampicillin resistant gene. We will see the other features of PBR322. As I mentioned, it is a size is 4363, 4363 base pairs, and it is having 2 selection markers, ampicillin resistance gene and tetracycline resistance gene. Ampicillin resistance gene is a beta lactamase, and which converts the penicillin into the penicillinic acid, so that the vector containing transformants grow in the presence of media containing the ampicillin. So the vector containing E. coli can be selected. Media is supplemented with the ampicillin. Even the transformants can be scored in the presence of tetracycline. As I told you that there are 14 sites, the 9 sites from the tetracycline gene and the 5 sites from ampicillin gene. These unique sites, unique means only single site is available in the construct. These 14 sites can be used for the cloning of your gene of interest. Cloning strategy is only one site you have to use. That's why it is called as unidirectional cloning. So only one site. Then how much you can, what is the size of maximum size of fragment you can clone by using this vector? You can clone 5 to 6 kb of insert, because whenever plasmid total size is reaching 10 kb, beyond 10, it is unstable. So already the PBR322 vector size is 4363. Hence, we can clone another 5 to 6 kb of insert into this vector. Selection, whenever you are cloning your gene of interest into the one of the antibiotic selection marker, so the transformants means only vector is resistant to both antibiotics, but a vector with the insert is resistant to one antibiotic and sensitive to one antibiotic. So if at all you use the restriction site from the tetracycline to clone your gene of interest, the recombinant is sensitive to the tetracycline and resistant to the ampicillin. If you use the ampicillin restriction site, it is nothing but PST1, then the transformant, which is containing the vector or recombinant, then that is sensitive to the ampicillin and resistant to the tetracycline. So whenever you're cloning your gene of interest into the antibiotic selection marker, then it is losing its ability to score on the antibiotic. So this kind of selection is called as a insertional inactivation. When you clone your gene of interest into the antibiotic selection marker, then that gene is disrupted and the recombinant is sensitive to the particular antibiotic. So this process of selection is called as insertional inactivation. So how many copies of pBR322 can be there in the each ecocell? Maximum of 15 kb. So it is called as a low copy plasmid. And as you all know that origin of replication is necessary for the autonomous replication, it is there. And the repressor of primer is required for the maintenance of the copy number. And PBR322 was used to screen the DNA fragments with the promoter, right? Then also the PBR322 was used to express the chemically synthesized gene, for example, somatostatin. Somatostatin was the first expressed in the clone in the PBR322 and expressed in E. coli. So these are the 10 points of PBR322. In the next one, we will see the limitations of the PBR322, even though it is a first cloning vector. And the first and the foremost is the screening is very lengthy and laborious. It is a two-step process. First, you have to screen the transformants on a media containing the non-used antibiotics. For example, there are two selection marker. One is the tetracycline and ampicillin. If you use the restriction site of ampicillin to clone, then the ligation mixture and transformants are scored on tetracycline. If you use the tetracycline restriction site, then you have to transformant as need to be selected on ampicillin. Then from there, the transformant which grow, then again you have to do the replica plating, then you have to take to the sensitive one. So it's a lengthy one. So the limitation is, again, you can clone 5 to 6 kb. It cannot be used for the genomic or CDN libraries. Only for cloning purpose you can use. And unidirectional cloning, again, you are using only one enzyme for cloning. So orientation, you can get the reverse orientation also. This is a one orientation. Since there is a possibility to have one more orientation, because so it can fit into the either orientation. So these are the limitations of the PBR322. And it is not ideal for expression of proteins. This is not an ideal vector for expression of any protein. So this is all about PBR322. If at all you like it, this video, try to subscribe my channel, that is Dr. K PREM-PRIMER, for regular updates. And if you want to you do share with your friends, and try to give thumbs up if you like it. And if you have any doubts, please write to me through the comment section. I'll get back to you as soon as I see. Thank you all. See you again with one more video. Until then, bye bye.